In this video, we're going to be highlighting glycolysis, which is glucose metabolism. And we've started off by drawing the Fisher projection of glucose, where we draw on the left side a symmetrical distribution of 2 hydrogens, an alcohol, and 2 hydrogens. And then on the right side, we fill in the partner. If we have a hydrogen on this side, we need to have an alcohol on the right. And we follow that trend all the way down. Now, our mnemonic to remember how we go from a Fisher projection to a Hayworth projection is drool, which just means down on the right. So any substituents on the right are going to be pointed downwards, and up on the left. So any substituents on the left are going to be pointed upwards on the Hayworth projection. So that's what we see being represented here. This alpha designation is dictated by the anomeric carbon, and more specifically based on the orientation of this alcohol around that anomeric carbon. So it's pointed downwards, that is in the alpha configuration. So we're going to be using our Hayworth projection to discuss the glycolysis pathway, where our first step is one that is irreversible and is catalyzed by an enzyme called hexokinase. That prefix hexo is because there are 6 carbons, but the kinase tells us about what type of reaction is taking place. And kinases should trigger the thought that phosphoryl groups are moving. In this case, we're going to see ATP being used to form ADP so that a phosphoryl group can get added to glucose. And so we see that the phosphoryl group is being added to carbon 6 on our glucose molecule. So carbon 6 is there, and including our phosphoryl group now. So the overall structural change here is that we have now added our phosphoryl group that was once an alcohol. And this molecule is no longer just glucose, it's glucose 6-phosphate. Step 2 is one that is reversible. And its name is also going to tell us about what is taking place here. So the enzyme at play is called hexoisomerase. And so the term isomer is telling us that there is going to be a product that is going to have the same chemical formula as our glucose 6-phosphate, but a different structural arrangement. And so that difference is going to lie in the ring structure, where here we have a pyranose, a 6-membered ring, and down below we're going to see a furanose, or a 5-membered ring. The number of carbons, though, is going to stay the same, where now we have carbon 1 here. And the orientation of these substituents is also going to be maintained, where at carbon 2, we have the alcohol pointed down, carbon 2, the alcohol is pointed down. So we're going to orient the alcohol at carbon 3 upward, carbon 4 is going downward, carbon 5 is attached to carbon 6, that is attached to our phosphoryl group. So we've gone from our 6-membered ring to a 5-membered ring, and that is our key chemical change here. So now we have fructose 6-phosphate. Our next step is another irreversible process. And this is going to be a reaction catalyzed by phosphofructokinase 1. Also called PFK1. And this kinase is telling us about its role in moving phosphoryl groups around, in this case from ATP to ADP, so that we can add a phosphoryl group to our first carbon. So we previously had an alcohol at this position, and now we're going to see a phosphoryl group to phosphate molecule. So our overall change here is the addition of this phosphate group at carbon 1 to give rise to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate is acted on reversibly by the enzyme aldolase. And the name aldolase is giving us an idea about the mechanism involving aldol condensation. And this step is a little bit unique from what we've highlighted so far, where we have our 6-carbon species that is going to be broken down into 2 3-carbon molecules. And that break occurs at these green lines, where carbons 1, 2, and 3 are going to be used to form the molecule dihydroxyacetone phosphate, and carbons 4, 5, and 6 form glyceraldehyde 3-phosphate. And the mnemonic that we've highlighted is that the breakage points look like a phase clock set to 130. And if we go in the clockwise direction, we meet D first and then G. So at 130, meet Donald Glover. So we'll start off by drawing our dihydroxyacetone phosphate. And so the way that I remember this molecule is that we have 3 carbons. And the second carbon is going to be one that has a carbonyl, and the third has our alcohol. The reason why I remember the second carbon has the carbonyl is because if we look at this highlighted region, that looks like acetone. So that's how we get our dihydroxyacetone phosphate. Our second product is our glyceraldehyde 3-phosphate. So writing down our phosphoryl group first, we're going to have the same 3-carbon type of structure, where at carbon 2, we have an alcohol, and at carbon 1, we have our aldehyde. And this is our glyceraldehyde 3-phosphate molecule. The last step of our preparatory phase is catalyzed by the enzyme triose phosphate isomerase, which is abbreviated as TIM or TPI. And so the word isomer is indicating that we're going to have the same chemical formula, but a different arrangement. And that arrangement is going to involve converting dihydroxyacetone phosphate into a second molecule of glyceraldehyde 3-phosphate, at least in this forward direction of glycolysis. And so that's going to just be reflected as 2 of our glyceraldehyde 3-phosphate molecules. Carbon 2 has our alcohol. Carbon 1 is where we see the aldehyde. And so that is, glyceraldehyde 3-phosphate. And there are now 2 molecules. So essentially, we've seen a change at carbon 2 and carbon 1. Our 2 molecules of glyceraldehyde 3-phosphate are going to kick off the payoff phase, where we see a lot of moving parts in step 6. So I'm going to draw longer arrows than usual. And we're going to see that this is carried out by glyceraldehyde 3-phosphate dehydrogenase, also called GAPDH. And so this dehydrogenase term should suggest that there are hydrogens moving around here. And so there's the first moving part we're going to write down are 2 phosphate molecules are going to be used during this reaction. We're also going to see 2 NAD molecules included. And these are coenzymes that are going to be used to make NADH and protons, or to release these species. And so the resulting product here is going to be our 1,3-bisphosphoglycerate. So we're going to add a phosphoryl group here in place of our hydrogens. So we'll have 2 of these molecules. Where we have our phosphoryl group maintained at carbon 3. So this is carbon 3, 2, 1. Alcohol will be maintained at carbon 2. But now, instead of just having an aldehyde at carbon 1, we have a second phosphoryl group. And so that is our overall chemical change here. And this molecule is called 1,3-bisphosphoglycerate. And so not only have we created a bisphosphate molecule that's going to be very favorable in downstream steps, but we've also synthesized NADH, which is a wonderful electron carrier that we're going to highlight in detail when we talk about electron transport. So 1,3-bisphosphoglycerate gets recognized by a kinase. And this kinase is called phosphoglycerate kinase. And this is not to be confused with phosphoglycokinase. This process is going to be our first observation of substrate level phosphorylation. And what I mean by that is that we're going to take one of the phosphoryl groups from our substrate, in particular this one, this phosphoryl group, and donate it to ADP in order to form ATP. So the resulting product, and so for sake of consistency, let's also highlight the kinase is what we're using as our clue to know that we have phosphoryl groups moving around. So we have two of our three phosphoglycerate molecules that are going to form. And so we have a carboxyl group there that is left behind. And so that is our key chemical transformation, is that instead of our phosphoryl group, we have just a carboxylate, so an oxygen that's left behind there. And so we're going to call this 3-phosphoglycerate. The next reaction is catalyzed by a mutase called phosphoglycerate mutase. And this is a reversible reaction where there's going to be a change to the molecule, where specifically the phosphoryl group at carbon 3 and the alcohol group at carbon 2 are going to switch places. So we're going to synthesize 2-phosphoglycerate. So we'll make two of these molecules in which the alcohol is observed at carbon 3, and I'm actually going to adjust how I have this carbon backbone orientation just based on where my room is. So I'm going to have my phosphoryl group at carbon 2. So we have two main changes here, where we have our alcohol at carbon 3 and our phosphoryl group at carbon 2. So this is a really similar molecule to what we had before, but we're going to call this 2-phosphoglycerate because the phosphoryl group is now at carbon 2. Our next step is a dehydration that is catalyzed by enolase. And so we're going to see the loss of water, 2 molecules of water. And so this, as the name suggests, is going to involve an alkene. We're going to form an alkene here, where we have 2 molecules, where carbon 2 is going to be here. We have our phosphoryl group. And carbon 3 is still going to have the carbonyl that is part of the carboxylate. So this molecule is called phosphoenol pyruvate. A very high energy molecule. At least for hydrolysis. And our last step is one that is irreversible, that we're going to see catalyzed by the enzyme pyruvate kinase. And kinase means we got phosphate moving around. So we'll see 2 ADP molecules being converted into 2 ATP molecules, which suggests that we have another substrate level phosphorylation process in which we're going to see our final product of pyruvate, where we have a carbonyl at carbon 2 and our carboxylate at carbon 3. Excuse me, I've numbered that incorrectly. We have carbon 1, 2, and 3 here. And so this is our pyruvate molecule. And that is how we go through the glycolysis process, where our last chemical change here was we've lost our alkene. It's now being used to form our carbonyl at position 2 instead of our phosphoryl group. And that's our glycolytic pathway.